I'm Dr. Laura Glenn. I'm one of the naturopathic physicians here at the office. And tonight we're going to talk about GI health. It's microbiome and the GI health, but I'm actually kind of going to give you like a crash course in gastroenterology. Like just really brief, giving you a sense of how does the GI system function, how does it work when it's working correctly, what it's supposed to be doing, and then what happens when it starts working incorrectly and when things get out of balance, and then how can we kind of help rectify that situation, make things a little bit better. So, just to start out with, in case you're not familiar with naturopathic medicine, I'm not sure, everyone here is probably already a patient or is a friend of a patient, but really quickly, to give you a sense of what it is that we do and what our training is, we're trained to kind of bridge the conventional primary care world and all of the alternative stuff, like all of those modalities. So we're really trained to kind of integrate both of them and to act as a primary care provider, being able to diagnose and manage common conditions, including medications when needed, but using more of a holistic mindset. So rather than just getting to a diagnosis and saying, well, here's a drug, we get to a diagnosis and say, well, how did you get to that point in the first place? Because everyone's process is a little different. So how do we find what's the root cause of yours and use all of our natural therapies to try to treat that, making the body healthier as a whole, stronger. So really, same kind of first 2 years are the same as conventional medical school. It's really the last 2 years where we incorporate all of the modalities. So the acupuncture, herbal medicine, more of the nutrition, all of that. So just to give you a sense. Okay, so let's dive in. So what is the GI tract? It's technically, everything inside of the GI tract is outside of us. It's like this long tube from mouth to bottom, that anything that goes in there, until it gets passed through all of those individual intestinal cells, it's still technically outside of us. I just think that's a really cool fact, so I always throw that in there. It's really long, actually 20 feet if you were to stretch everything all out. So if you think about how much surface area that is, it's really one of our largest organs in our whole body if you were to take the entire thing. It starts with the mouth and esophagus. I don't have a pointer on that, but you can kind of see mouth to esophagus, it goes into the stomach and then works our way on down. And we also include like the liver, gallbladder, and pancreas. They're called accessory organs, meaning nothing's really passing through a tube of them, but they have a lot of play with what's going on with our digestion. So, what does the tract do? Digestion, absorption, assimilation, meaning are we taking things actually across the cells and into our body, and then excretion, getting rid of the waste. So why is this important? We always hear, well, you are what you eat. It's really more about you are what you absorb. You could be eating a perfect, great diet, but if your GI tract is not healthy enough to actually take your nutrients up, it's kind of meaningless. So in addition to that aspect of it, we have to think about what's going on with the nervous system. We've all had that experience of butterflies in the stomach and feeling like a nervous tension there. There's actually a whole enteric nervous system that's happening in our intestinal tract, and it does interface with the brain, the central nervous system. And there's so much of a connection there that we actually are starting to refer to this as the second brain. So saying that you have a gut feeling about something literally is kind of right. Really, just a quick interesting fact in terms of the interface between the brain and the gut. For those of you that remember your anatomy or learned your anatomy, one of the cranial nerves that goes to the digestive tract is the vagus nerve. We always think about that as leaving the brain and heading that way, but they're finding more and more, the more they look at it, there's actually fibers backtracking up, going from the gut back to the brain along that same track. So there really is quite a kind of two-way street there. It's not all just the brain telling the gut what to do. There's a lot of feedback about what's happening in the gut, telling the brain, what do we do here? And the gut's our first line of defense. You think about how much we take in our mouths in terms of breathing, swallowing food, touching things with our hands, rubbing our mouth, all of that. The gut has like kind of our primary immune system located in there. So everything that we're coming into contact with in the outside world, it has to decide is this friend or foe, and then either kill it or let it live. Okay, so real briefly, physiology of digestion, because a lot of what I work with is people that have kind of functional gut disorders, so irritable bowel syndrome, things like that. When we think about what's going on there, we have to really think about how are you supposed to be digesting your food, and a lot of times it starts with not chewing well and not chewing slowly enough and eating slowly enough. So chewing actually starts the breakdown of our starches. We produce something called amylase, which just gets that process going. Starches are kind of long and branch chain looking. We have to get them all broken down to individual little carbohydrate molecules. So we start there. everything then goes into the stomach. We produce pepsin, which kind of starts breaking down our proteins. Again, same idea. We start out with these really long molecules, and by the end of the digestive process, when we get, and I won't even call it the end, but when we get to the small intestine, where everything's supposed to be absorbed and taken up, if things aren't broken down well by that point, you will not absorb them well. They're just going to get passed into the colon. So pepsin breaks down the proteins, makes some stomach acid. We have to have that acid to activate the pepsin and start that protein breakdown. And then we have a lot of muscular contractions. You can see, I don't know how well you can see this. I think it's cool though. There's like 3 different layers of muscles. They're all going in different directions. So everything's all just getting squished up and kind of emulsified, liquefied. Okay, so after we get out of the stomach, it goes into the small intestine. This is where carbohydrates, proteins, and fats are digested and absorbed, along with electrolytes, vitamins, water. Small intestine is a really big deal. In terms of carbohydrates, we started talking about the breakdown that happens with the saliva in the mouth. But after everything kind of gets into the small intestine, the pancreas, which is kind of located here across the middle, it dumps pancreatic enzymes, kind of a mixture, a concoction of things that are going to break down all of these food particles into smaller pieces. And intestinal cells secrete some more. So particularly, we think about people with lactose intolerance. There's enzymes that are actually made on the tips of all of the little intestinal cells that break down lactose, milk sugar, into more simple particles that we can then absorb. So there are some enzymes actually being made by the cells of the intestines. It's not all just coming from the pancreas. Okay. Proteins, again, pancreas secretes some things that break proteins down a little bit better. Same thing, lining of the upper small intestine secretes more enzymes that break them down further. Basically, the concept I'm trying to get across here is that we have to make sure that the pancreas is functioning effectively and that the cells in the small intestine are healthy enough to secrete their enzymes too. There's a lot of different steps that happen in digesting our food that we don't think about, and when one of them starts breaking down, a lot of things can kind of go sour. So, all right, fats. So most people are familiar with the idea of the gallbladder. It kind of sits next to the liver. It stores bile. Bile emulsifies our fats. That's going to make it easier to absorb later. Because think about fat separates from water, right? Like if you've ever put oil and vinegar together, they just separate out until you shake them up real good. Same idea. We need bile to help mix everything all together. Okay, absorption. So most of this actually happens in the second part of the small intestine, and these are those little finger-like projections that I was talking about. So all of your cells have all these little fingers, which I think is kind of cool. And so a lot of the enzymes get made there, and then a lot of the digestion happens across that. So we'll talk more about what happens when these barriers kind of get broken down or not as efficient as we want them to be. So after we get out of the small intestine, we go into the large intestine. At this point, most of our food and nutrients should have been absorbed. The large intestine is really just about reabsorbing water, salts, and moving the waste along so we can excrete it. But this is where our gut flora is supposed to be living. This is where the bulk of it is residing, is in the large intestine, or the colon. Actually, I think I come back to that idea, but one thing I will say is if you think about that process of how everything's supposed to be broken down well and absorbed well in the small intestine, if that process is faulty somewhere, and you're not breaking everything down well, so it's not really being well absorbed, part of that food gets put into the colon. I mean, it's not like big chunks of food. It is broken down to some extent, but it wasn't small enough to get absorbed. When it gets to the colon, that's when we can start getting into a state of dysbiosis because we're leaving those food particles to feed bacteria, both bad and good. Bacteria are naturally going to have some foods that they like to metabolize, but when we're leaving a lot of extra stuff around by not absorbing it and digesting it well, that's when we can get into more of a dysbiosis state. So, healthy digestion in stools. Everyone pretty much knows this stuff. This is like what we're aiming for. So at least one bowel movement a day that's well formed, doesn't smell terrible, doesn't have blood or mucus, doesn't have undigested food in it other than corn, nuts, things like that that no one digests well. We should aim, I mean, no one really checks this out unless they're nerdy about the GI tract, but bowel transit time really should be between 18 and 24 hours. If you ever wanted to check what that means or how long it is, you could either eat corn and see how long it takes from lip to pass, or you could do something like activated charcoal. It's going to turn the stool dark. So you could see how long is it taking me from when I put it in my mouth to then coming out. If it's very slow, slower than 24 hours, there's some issues going on with not having a fast enough transit time. Or if it's coming out quite fast, you're probably not absorbing everything as well as you should, because it does take a certain amount of time for everything to work its way through the system and get absorbed in an ideal manner. And then no gas, bloating, no indigestion after meals. We don't really want those things. Okay. So now the fun stuff, stuff that I like. Talking about the microbiome, like all of the bacteria that are living inside of us. I have interesting factoids for you because I think this is amazing. So we have 2 to 3 pounds of bacteria living in our gut. That's 100 trillion organisms, and they're finding it's between 15,000 and 40,000 different species, completely different types of bacteria. We really have no idea what all these bacteria are or even what they do. We just know that they're there, and they must be important to us. So collectively, the cells of our microbiome exceed our own human cells by a factor of 10 to 20 to 1 of ours. So there's literally more foreign things living in us than there are of us, which I think is kind of amazing. And then the gene pool exceeds our own genes by 100-fold, so 100 to 1. So we have to think about how much of an impact is that having on our own health. There's that much diversity in us, or there at least is supposed to be. And what is that doing to us? So what do these bacteria do? Some of them actually do function in a manner that is crucial for us in terms of producing vitamins, nutrients, and helping us absorb a little bit better certain things. Like vitamin K, we think about that as being, pretty essential to us, and we get that from bacterial. They also break down drugs and toxins. So if you don't have a robust flora in the gut, you may not be breaking things down as well as you should, and potentially could be, reabsorbing some of these toxins or these drugs and recirculating through the system until they finally get broken down well enough to be excreted. And then they regulate a lot of aspects of our immune system. So they protect us from infections and chronic inflammation, and possibly many immune-based disorders. So autoimmunity, eczema, allergies, things like that. They're finding more and more that there's a big connection between having a good, robust gut population and what's happening with our overall health. So where do we get all of these bacteria? We begin colonization with birth. I have a little note about vaginal versus cesarean birth. So the way that humans for millennia have come into existence is passing through the birth canal, the vaginal canal. And we pick up bacteria from our mothers as we're doing this. When babies are born C-section, they don't get that initial inoculation of bacteria. So their gut bacteria actually looks quite different than a baby that was born vaginally. So the C-section babies tend to be missing a lot of the bifidobacteria species, and they're actually finding that the C-section babies tend to have a lot more of the bacteria that was like more skin bacteria of the healthcare workers and of mom, because that's really their first exposure, is they're exposed to skin, like what we're having living on our skin. Okay, so then during the first year of life, it looks a lot different than an adult, but after about 1 year old, the gut colony tends to look more like an adult. So it's really everything that they're coming into contact with as well. Babies are always on the ground, they're picking things up, sticking things in their mouth. They're getting exposed to a lot of different bacteria that way. The idea is that once we're adults and fully developed, our microbiome should more or less stay the same. It doesn't really happen in today's day and age, but that was the thought. So, the hygiene hypothesis. Why are we so sick all the time now? Why do we have so much autoimmunity? The idea is that developing infants need exposure to a wide variety of organisms to develop a healthy immune system, because all of these different bacteria help train our immune system to learn friend from foe. It develops a tolerance, and it says, okay, well, these things are all foreign, but they're not harming me. These things are foreign and they're harming me. I need to attack those things. It has to kind of learn what's a problem and what's not. When it doesn't learn what's a problem and what's not, it doesn't properly develop and learn how to function well, and that's when we can start getting things like autoimmune conditions, asthma, eczema, things of that nature. So the idea is in countries with high levels of hygiene, are we being exposed to enough microbes? Are we getting enough colonization? And there is correlation in terms of, and I didn't write this on there, but we do see that in more industrialized countries we have higher rates of asthma, allergies, and autoimmunity. We don't see that as much in less developed countries that don't have quite as good sanitation practices. Obviously we want good sanitation, but we need to find a balance. Okay, so in terms of how can we achieve that balance, thinking about antibiotic use is probably the #1 thing that we should be thinking about as a society and where we're going. So human and animal studies have both shown that even one antibiotic treatment leads to decreases in our good bacteria and increases in potential pathogenic bacteria. So like C. difficile, some yeast species. Stress also will kind of knock down our immune system definitely, but also our good bacteria and increasing pathogenic bacteria. And why that exactly happens, I don't think we have good definitive answers. I have a suspicion it has to do with our cortisol balance and how the adrenals are impacting the immune system as well. But how long does it take to recover? So after antibiotic treatment, how long does it take to get your good bacteria back to robust levels? So studies have shown that most types of the bacteria will return to pre-treatment levels by 4 weeks, at least after ciprofloxacin. That was the study that they did here. But a few types of bacteria they looked at did not recover even 6 months later. They've also shown that some types of bacteria remain disrupted up to 2 years after a round of clindamycin. So it's not that easy to get the good bacteria back. We also have to talk about antibiotic resistance, because this is one of my things that I'm just very passionate about. So the more antibiotics we use, the more resistant strains we can pick up, because if a bacteria has a gene that's resistant to an antibiotic use, it's not going to get killed by whatever antibiotic we give. And bacteria do this really interesting thing called gene sharing through plasmids. They don't even have to be the same species of bacteria. It can go to a totally different bacteria species and share its genetic material and give it this genetic resistance to the drug. So if you had, I don't know, I don't have a good example, but if you had one type of bacteria that you had an infection of, like a urinary tract infection, and it was resistant to one drug, you used it, you killed off most of what was there, but one of the bacteria survived because it was resistant. It can then go to other bacteria and transfer those genes, and we can pass it on, and it gets to be a huge big mess. So really, we just need to be using antibiotics more judiciously. We need to really use them for situations where we can have serious complications or death from. There are no help in viral illnesses. When people have a common cold and want treatment for that, giving an antibiotic is not the answer. It's just, again, it's going to contribute to antibiotic resistance developing. I don't really think we should be using antibiotics in livestock as much as we do, unfortunately. It's pretty common practice in conventional agriculture. Makes the animals grow faster. But again, we're contributing to more antibiotic resistance developing, and that does spread from animals to humans, and then from the human farmers that work with the animals into the surrounding population. And do we really need antibacterial soap? This drives me crazy. Soap is soap. You wash your hands with soap, you're getting rid of the dirt. You don't need antibacterial soap. Okay. And then we should be repopulating with probiotics after we take our antibiotics. Try to get those good colonies back up. All right. Now going into like the more interesting stuff. I just have to give the disclaimer, it's not medical advice, it's educational information. Talk with your doctor, your healthcare provider about what might be right for you in terms of treatment. So choosing a probiotic. I like to start with the idea of the prebiotic debate because there's a lot of different thoughts on it. Prebiotics are fibers that are like food for probiotics, for the good bacteria. So they're inulin, oligosaccharides, lactulose, resistant starches, like FOS. Most people, if you've looked at the back of a probiotic label, you'll see fructooligosaccharides, FOS. The idea is that it helps us grow more lactobacilli and bifidobacteria. But it can feed other bacteria as well. So one thing that I would say about that is that I've seen people who have pretty bad dysbiosis take a probiotic with prebiotics in it, and they feel worse. So very possible that the prebiotics are feeding the bad bacteria because we haven't identified what those bad bacteria are and killed them off or gotten rid of them. Or for people that have small intestinal bacterial overgrowth, SIBO, again, prebiotics not such a good idea because if you haven't killed off those bad bacteria that are living in a place they're not supposed to, which we'll talk about again, we'll get there. But again, we don't want to feed bad stuff, we just want the good stuff there. So just a thought to consider. Generally we want colony-forming units, CFUs, in the billions with a B, not millions with an M. So we want high potencies. We want multiple strains of probiotics in the product. So when you're looking at the label, different ingredients, if you will, different names of the different probiotics. And I always recommend taking them with food. There's a lot of probiotics that are labeled to take on an empty stomach. The literature that was suggesting that it's good to take it away from food has been outdated for like 10 years. So just take it with food. You'll get more of it through into the small intestine and it'll then get into the large and better colonize. Okay. Indications for probiotic use. Again, during and post antibiotic treatment with chronic GI issues. So in that regard, like irritable bowel syndrome, irritable bowel disease, food allergies. The probiotics, again, they help maintain a good gut barrier. They keep our GALT, which is gastric-associated lymphoid tissue. It's like the local immune tissue in the GI tract. Keeps that strong, decreases inflammation, and improves processing of antigens, meaning like kind of immune proteins that are coming through. Good in autoimmune conditions because, again, we think about there was likely some dysbiosis before the condition developed, so we're again trying to develop more tolerance of the immune system. And then there are long-term benefits even in healthy people. Better hormone balance, improved immune system, response to infections. In terms of hormone balance, that's one thing that I like to talk about because most people aren't familiar with how that impacts hormone balance, what that bacteria is actually doing when we talk about hormones. But when our liver is clearing excess hormones from the body, so we're always making more, we have to get rid of some of it somehow. The liver puts a compound on there to make it more soluble in the feces, to get it out so that it's not reabsorbed into the blood. That compound has a little bond that can be cleaved by an enzyme produced by unfriendly bacteria. The enzyme is called beta-glucuronidase, if you were curious and wanted to look it up. But anyways, if you have too much unfriendly bacteria making more of this enzyme, you're going to cleave that bond, the hormone's not going to be soluble in the stool anymore, and it's going to be reabsorbed back into your bloodstream and into circulation. So we really want a good, healthy population of bacteria in the gut so that we are processing and eliminating our hormones well and they're not constantly being reabsorbed. Yes?
Is that specific hormones or all of the hormones?
That's a really good question. I'm most familiar with it with estrogen, but I would guess that it's pretty much all of them. I mean, they've all got the same steroid backbone. Do you know, Dr. K?
I missed
The beta-glucuronidase issue, is that just estrogen or is that all hormones when we're talking about recirculation from bad bacteria?
Mostly estrogen.
Mostly
It may do a tiny bit of the rest, but mostly estrogen.
Yeah.
So does that imply that the studies are done mostly on estrogen?
Probably. I don't know. That's what I'm most familiar with.
I don't mean to torture you, I'm just trying to get some clarity.
It's a good question. It's a good question. And more studies, especially about the microbiome, more studies are being done all the time. It's kind of a really exciting time in the study of what's happening in our gut. So I bet we'll have more answers in a few more years. We just need to wait for science to
Check
a little more.
Yeah. Thank you.
You're welcome. Okay, so the idea of getting our probiotics from food, if you're not going to get it from a pill, a lot of people want to get it from food. Conventional yogurt, it's really not that good of a source. It just really isn't. The colony counts are low. It's not fermented in the same way that homemade yogurt or old-fashioned yogurt is fermented. So if we're going to think about getting probiotics from food, I would go with the traditionally fermented foods, like kefir, kombucha, which is like a fermented tea, miso, tempeh, kimchi, homemade yogurt, old-fashioned pickles like homemade lactobacteria fermented pickles, homemade sauerkrauts. Things that are actually fermented by bacteria, not by vinegar with some bacteria thrown in for good measure. If this is something that interests you, I have an awesome book to recommend. It's called Wild Fermentation by Sandor Katz. It's great. He even has like recipes about how to try these things at home for yourself. Okay, so now kind of moving into different health conditions and what we do as naturopathic doctors. So irritable bowel syndrome, most people know what this is, so I'm not going to take a whole lot of time going over the symptoms. I would just say that conventionally they don't have a lot of good solutions for this. It's okay if it's diarrhea predominant, we stop the diarrhea. If it's constipation predominant, potentially we give an antidepressant. That seems to be their answer lately. The reason, again, thinking how much of a connection is there between the mind and the gut, how much serotonin is actually produced in the gut, it's a very large amount. So I can see where they're going with the antidepressant use, but again, it's not addressing the root cause of why we're having the issues in the first place. So as naturopaths, where do we start? So a high percentage of these patients have dysbiosis, whether that's in the colon or in the small intestine. So I mentioned SIBO, small intestinal bacterial overgrowth, and that's a very real issue that's really become more well known, I would say, in the last 5 years or so. And it's where instead of the bacterial population residing entirely in the large intestine, we actually have some backflow, and they're kind of migrating backwards into the small intestine. So when they get into the small intestine, if you think about, remember where all that digestion's happening, all the food's in there. So there's lots of food for them to grow and proliferate and you know, thrive, really. So they can create a lot of gas, bloating, inflammation if they're located in the small intestine. We really don't want them there. So there's specialty lab testing that looks for pathogens and dysbiosis in general. There's two main types that we do. One would be a stool test that's looking at ratios of good to bad bacteria, looking for any parasites, anything like that. If we're more suspicious about SIBO being an issue, that's actually done through a breath test. So you take a solution that the bacteria would like to eat and then thus create some gas from, and its byproducts of metabolism, and then we collect the breath in a bag and it gets sent off, and they look at, well, how much methane and hydrogen are actually being made. If it's made in a certain amount of time, it's coming from the small intestine, there's bacteria there. So when we find out what the imbalance is, then we need to treat it. So whether that's in the colon or the small intestine. Additionally, with irritable bowel syndrome, these patients are more likely to have food intolerances or food allergies. So we can test for those as well. If there's foods that you're eating in your diet that just aren't working for your system, even if they're healthy, it's going to create more inflammation. There's the cortisol serotonin imbalance. So working on the stress reduction is one thing that we work on. And then we can use calming herbs for the intestine, things to kind of just make everything settle. Really, I look at that more as a it's palliative, it's helpful, but it's really probably not addressing the underlying cause. So we really need to get at what's happening there with dysbiosis, with foods, with your digestion even. Okay, before I move on, questions about IBS? No? Okay. So next condition, reflux. Everyone knows what reflux is, or most people do. So some things that I like to point out that some people aren't familiar with are symptoms like a chronic cough lasting more than 8 weeks that's unexplained, feeling like a lump in the throat or more hoarseness, kind of a bitter or sour taste in the mouth. So what's happening with reflux? When we've got food in the stomach, things are getting all mixed up. If the lower esophageal sphincter, which is like right there, there's a muscle that goes all the way around that, that's a circle. If that does not close down tight, as everything's pushing around in there, you can imagine as it's being pushed, if there's not a closure, things are going to pop back up, including the acid from our stomach. So why can this happen? Why is that sphincter relaxed? H. pylori, which is a bacteria that can live in the stomach despite the insane amount of stomach acid, it actually thrives there. If we have H. pylori infection, that can cause that. Or just not making enough stomach acid. It's counterintuitive because you would think if I'm having reflux, I must be making too much. But if you're actually not making enough, you're not going to get the trigger, the signals to actually close down on that sphincter. So you could be making too little, but the sphincter's not triggered to close because it's not enough. And then when it's, grinding things away, the little bit that you are making is going to backflow. Other things that can cause reflux are a hiatal hernia, so where the top of the stomach kind of slips through the diaphragm, the hole, or increased abdominal pressure, pregnancy, obesity, really heavy meals, where there's too much pressure on the stomach, it just creates more backflux. And then there's some common food triggers. Most people who have reflux have already been told about these things, the coffee, tea, citrus. some that I like to point out that people aren't always familiar with are the onions, chocolate, mints. Then there's a whole host of problems that are associated with uncontrolled reflux, when you can't get it under control. Conventionally, what they tell you is eat smaller meals, don't eat late at night, don't recline too far, keep the head of the bed elevated, stay upright after you eat, and then they use a whole host of antacids, basically, things that block either histamine or acid. Okay, proton pump inhibitors. I want to point this out because I see lots and lots of people that have been on proton pump inhibitors for years and years, thinking that this is great, because it is they're controlling their acid. They feel, I don't have the reflux anymore. However, with proton pump inhibitors, we're not making as much stomach acid as we should. When we don't make as much stomach acid, we can't absorb our minerals as well. So we're at an increased risk of bone fracture because we're not taking up the calcium that we should and mineralizing the bones. We also have increased risks for dysbiosis because we don't have that good level of stomach acid to kill off all of the pathogens that we're coming in contact with. We're at increased risk for food sensitivities. And they've actually found an increased risk for pneumonia, surprisingly enough. Again, I think it's, this is kind of our first pass. It's what we're getting into the system in terms of bugs, what we're exposed to with pathogens from the outside world. That stomach acid's supposed to kill things off, bad things. But if you've got some serious problems going on in the esophagus from the reflux, I do see, there's a place for PPIs. There's a place for those proton pump inhibitors short term to get everything under control while those tissues can heal up. PPIs can be weaned off of. It just takes some diligence and it takes some patience. I would really not recommend stopping them cold turkey. You'll get massive amounts of kind of rebound acid production and some more reflux. So it just has to be done kind of carefully, and I'd recommend talking to your doctor about it. And then some problems with antacids, so the things like Tums and Rolaids that we think of. Calcium is a common ingredient that's used in there to neutralize that acid, but calcium also stimulates gastrin. And gastrin is part of our acid production cycle. So when the buffering action of the calcium wears off, you're probably going to get a little rebound acid formation from that. So just something to think about. We really want to address the underlying problem. Why are you having the reflux in the first place, rather than just masking it? So again, things that we do. We look at food sensitivities. We try to decrease inflammation. We try to promote better digestion either with bitters or actually giving more acid if you're deficient in that. So doing it like a stomach acid challenge. We talk about that sometimes. And then using herbs that are demulcents, meaning that they're kind of wet, soft. They give kind of a good barrier to help coat the stomach and the esophagus while everything's healing up. Okay, so food allergies. So I've talked about this a couple times now. So when we talk about food allergies, there's IgE allergies, which are the anaphylactic type, like the kid who eats a peanut and swells right away. Those are usually found pretty early in life. When we talk about food allergies as naturopathic doctors, typically we're talking about IgG-mediated allergies, which are a delayed onset allergy, or you can call them a sensitivity if you want. They are mediated by the immune system, so they are technically an allergy, they're just not an immediate allergy. So IgG food allergies can cause a lot of different health issues, mostly linked back to inflammation. They can be tricky to figure out what is the food that's actually causing the problem, or even linking it to the fact that it is a food causing the problem, because it can take a couple hours to a couple of days before you actually develop the symptoms that the food is causing. Like I said, it's gradual, it's a delayed onset. So really anything that's linked back to inflammation could be impacted by food sensitivities, so migraine headaches, acne, GI complaints, joint pains, mood changes, respiratory irritation or mucus production. There's a whole host of other things, but those are kind of my starting points. So when we're thinking about might you have a food sensitivity or not, we can do things one of two ways. We can do IgG testing by blood, or we can do an elimination and challenge diet. So if we did an elimination and challenge diet, you basically remove common food sensitivity like causing agents from the diet. You eliminate them for a period of time and then systematically reintroduce one at a time, waiting a couple days to see what happens each time. Do you develop some kind of response? The issue is it's really hard to eliminate everything that might be causing a problem from your diet, and emotionally it can be really hard going through the process because it takes quite a bit of time. You have to eliminate everything for a couple of weeks, and then that gradual introduction, one thing at a time, several days at a time, it can stretch out for a couple months. So most of my patients really want more immediate answers. So most people end up opting to go for the blood test. And I just note that some companies seem to have better accuracy than others with the blood test. So
I have a quick question.
Yes.
Can you also do skin tests in addition to the blood test? What's more accurate?
So skin test would be more accurate for the IgE-mediated allergies, those immediates. So when we think about like environmental allergies, pollens, danders, things like that, that's a pretty good way of estimating those. If we were to do skin tests for delayed onset food sensitivities, it could take up to 2 days for something to happen. And because a lot of what's happening is really mediated by the gut mucosal system, we might actually not get a good positive response if we were to like put a scratch on the skin and dab pineapple antigen on it or something. So I really wouldn't recommend it for foods, but it's great for environmental allergies. Okay, so this is just an example of part of one food allergy panel that I've seen before that I've gotten back. And you can see, like, if you just take a look, like almond, asparagus, avocado, banana, all of these different foods that we test for between like 84 and 184 or more, it's really hard to eliminate all those things from your diet to systematically test one at a time. So a lot of times when folks do an elimination and challenge diet, they feel better, but they're still missing a couple of key components that they just didn't think to eliminate in the first place. So, like for example, mushroom came back as a 3 on this panel. That means there's a high level of antigen-antibody response happening. Who would have thought to eliminate mushroom from someone's diet during an elimination and challenge? You know, it's not common. We don't typically make that recommendation. So, but these can be very useful in terms of identifying what's creating inflammation in the system. I already mentioned this. And most people need guidance about what to eat when they do an elimination and challenge diet. So now kind of moving on to leaky gut, and I like this transition because a lot of times when I get back a food allergy panel, if there's massive amounts of foods that are coming back positive, we just automatically, we can tell, well, you have leaky gut. That's going on as well. We don't even need to do another test for it because you have so many positive foods that there's no way that that should be happening unless the gut barrier was compromised in some way. So, I don't know, I tried to blow this picture up. I don't know how good you can see it. But each of these like purple guys with the fingers on top are supposed to be an individual cell in the GI tract. And you can see these kind of lines connecting cell to cell. Those are the ideas of tight junctions from cell to cell in the GI tract. They're supposed to keep all of our cells nice and tight together. So providing a good barrier where every little molecule of food has to hit the edge of one cell and be transported across. They don't really show a good picture of that, but the idea of like a food hitting here and then being taken across and then put into the bloodstream. When we have a situation like leaky gut, where the intestinal barriers broke down, all of those tight junctions have been damaged in somehow. The cells are no longer held nice and packed tight together, they're kind of floppy, loose. They're still together, like obviously, because otherwise you'd be like, bleeding or something. They're still together, but it's not as good of a barrier as it should. So you can see that there's now an area for things out there, outside of us, in that center of the gut, to slip through the cells and get into the bloodstream. So that's what we start seeing when we see leaky gut. When people come back with these food allergy panels that have just massive amounts of foods that they're positive to, we think first about, well, how are they even getting those foods undigested to that point? There's usually some kind of breakdown in the digestive process. At least that's how I look at it. Because if everything was broken down perfectly well by the time it got here, there wouldn't be long enough chains of the proteins or the carbohydrates for the body to even identify as foreign and make an antibody against. So it wouldn't really come up on the food sensitivity panel. That's my personal theory. I see your face. Disagree.
I'm not disagreeing with you.
Okay.
It's
supposed to be broken down to really small particles by the time it gets to the small intestine. So there's something not happening when there's so many things being identified as foreign invaders, because the body won't make antibodies to something it doesn't recognize as foreign. So it shouldn't recognize a food particle as foreign if it's down to a simple protein or a simple carbohydrate molecule. Because we need those are our fuel. So it's when they're still a little bit longer that it says, well, this doesn't look right. So when we've got this kind of leakiness, things can slip through. That can be food, that can also be pathogens, bad stuff. So causes of leaky gut, because everyone wants to know, well, how did this start? How did I get this? Things like non-steroidal anti-inflammatories will irritate the lining of the gut and potentially do some breakdown. Alcohol can do some breakdown, certain medications, stress, inflammation, poor diet. Dysbiosis can be a primary cause. Eating the foods you're sensitive to can be caused by leaky gut, but it also will worsen the leaky gut too. And it's all a cycle. The more of this stuff you do, the more damaged it gets. Okay, so I'm going to kind of move away from that. But does anyone have questions before we go? No? Okay. So irritable bowel disease, like Crohn's and ulcerative colitis. I'm not going to spend a whole lot of time describing what they are. If you have it, you know. But they're chronic, painful. Typically, a conventional gastroenterologist just tries to stabilize things as best as they can. They usually also tell you to avoid raw fruits and vegetables. The fiber can be irritating for some people. What do we see as naturopaths in terms of what is happening in these kind of conditions? Dysbiosis, big cause of inflammatory bowel disease. Maybe not for everyone, but for most. I would say most of the people I've seen with inflammatory bowel disease, when you look at a stool panel, there's dysbiosis going on. Some strains of bacteria can produce toxins that actually damage the intestinal cells. And you can kind of read that. But it just, again, it breaks that barrier down. So then more inflammation, more problems like that are occurring and then getting across those cells. Okay, so we look for dysbiosis, we look for food allergies. We use alternative labs to look for the dysbiosis, because if you were to send off a stool sample to LabCorp and Quest, they're not going to give us those ratios of good to bad bacteria. They're going to look for the primary main things that they see